Forward. Hi, everyone. Thank you very much for being here today, and welcome to the second webinar in this inaugural AGM Preclinical Translational Science webinar series. Our speaker today is Dr. David Hackos. Dr. Hackos is currently a senior principal scientist at Genentech, where he has pain research. He obtained his BA in physics from Johns Hopkins University and his PhD from the University of California, San Francisco. And then he did his postdoctoral work here at the NIH, where he worked with Kenton Swartz on the mechanism of gating in voltage-gated ion channels. Dr. Hackos entered the world of pain research in 2001, and he started his work at Renovis, a small biotech company in San Francisco, where his group developed a series of TRPV1 antagonists. In 2006, he joined Roche in Palo Alto, where he worked on pain and inflammation targets such as P2X3, TRPA1, and KV1.3. At Genentech, his research focuses on the development of novel pain drugs, in particular by selectively targeting the Nav1.7 sodium channel. Two such selective Nav1.7 inhibitors developed by his group have been tested in early clinical trials. In addition to this research, his lab also studies the genetics and biology of peripheral neuropathies. His talk today is entitled Nav1.7 as a pain target and why selective Nav1.7 inhibitors have not yet shown efficacy in clinical trials. We're really excited to hear this talk, and I know many of you are really excited to hear the talk as well. So if you have any questions today, please enter them in the Q&A box throughout the talks, and we'll get to them at the end of the talk. So David, thank you so much for being here today. We look forward to your presentation. The floor is yours.
Okay, thank you very much for inviting me to give this webinar. I'm really excited to share some of our work on Nav1.7 and the drug discovery around this target. I'll start with a little bit of a brief introduction to Nav1.7. I'll tell you about our development as well as other companies' developments of selective inhibitors for the Nav1.7 ion channel. But I want to give special emphasis in my talk on why we haven't actually made a pain drug, by targeting Nav1.7, and why, for the most part, this target has been abandoned by the pharmaceutical industry at this time. Okay. So why were we initially interested in Nav1.7? Nav1.7 represents a really great potential pain target. And despite many, many years of work in the pain space by the pharmaceutical industry, it's still the case that chronic pain represents a major unmet medical need. In fact, as many as 50 million people in the United States suffer from chronic pain. And 20 million of those suffer from severe, debilitating chronic pain that really destroys their entire lives. This is, unfortunately, the current drugs, such as NSAIDs, opioids, and gabapentinoids, which work well in acute situations where you have acute pain or pain for a short period of time, have limited efficacy in chronic pain. And at the same time have significant side effects. So we are still very far away from having what we'd really like to see in medicine around being able to treat chronic pain. The pharmaceutical industry has focused on this problem with the desire to make better, more specific, and more efficacious chronic pain drugs. Now, Nav1.7 represented potentially a very exciting target that had the potential to enable a novel type of pain drug to be developed. And the reason we became so interested in Nav1.7 is because of human genetics work that was done in the first half of the year 2000. what was discovered was that individuals such as Ashlyn Blocker here, who was highlighted in the New York Times Magazine, individuals that lack a functional copy of Nav1.7, so they have mutations in both chromosomes that knock out the Nav1.7 gene, have no ability to sense pain at all. They have a condition called congenital insensitivity to pain, CIP. So they can literally stick knives through their skin. They will accidentally put their hand in boiling water. They will sometimes injure themselves, breaking a leg or stepping on a nail, and not realize that they've done that until they damage their leg more. They do have defects in olfaction. We know that Nav1.7 channels are expressed by olfactory neurons and are important in olfaction, but otherwise they are remarkably normal. They have normal cognitive function. They have normal touch sensitivity. They have normal autonomic function. So this got us very excited that there was a gene that could potentially be targeted, because it's an ion channel, that had this very, very specific effect on pain. At the same time, it was discovered that certain point mutations in Nav1.7 that increase the activity of the channel could also cause diseases with the opposite phenotype, that where the individuals affected have more pain, such as erythromelalgia or inherited erythromelalgia, paroxysmal extreme pain disorder, and small fiber neuropathies that are caused by point mutations in Nav1.7. So we had mutations that both caused loss of function and gain of function, and eliminated pain or increased pain appropriately, making it a very exciting target. So what is Nav1.7? It's a voltage-gated sodium channel. It's a membrane protein, a very complex membrane protein containing 24 membrane-spanning alpha helices that sit in the membrane in this way. The structure of these channels is such that you can think of these transmembrane domains as being in 4 similar but non-identical domains called domains 1, 2, 3, and 4. Each domain contains this purple region, which is a pore-forming part, and as well as a green region that represents the voltage sensing domains. And these domains come together, they wrap around each other to form the structure shown on the right, which is a 3-dimensional structure of the sodium channel. And here what you see is the pore-forming domains in purple form this central axis where sodium ions can permeate through the channel, and the green regions are 4 helix bundles that are flanking the pore-forming domain of the channel. Each 4 helix bundle has this series of positively charged arginine residues that are sitting in the membrane and are sensitive to the electric field of the membrane. And this is how the channels are able to monitor the voltage of the membrane and open and close appropriately depending on the voltage. That makes them voltage-gated ion channels. Now, there are 9 such voltage-gated ion channels expressed in mammals, including humans. And it's important to realize that for the pain drug that we were trying to make, we needed to inhibit specifically and selectively Nav1.7. There are already drugs that inhibit all sodium channels, such as lidocaine and novocaine. And when you apply these locally to the skin, for example, they are extremely effective in inhibiting pain as well as other sensations such as touch. So we know that inhibiting sodium channels could give rise to an extremely effective drug. The problem is that you can't inhibit sodium channels throughout the body because these channels are required for all of your bodily functions. And you would essentially die if you inhibited all of your sodium channels. So what we wanted to do was inhibit Nav1.7 specifically. And that was, at the time that we started these projects, that was the big challenge. Could we do that? And I should mention that the drugs that are non-selective, such as lidocaine and novocaine, block the channels by binding into the pore and plugging the pore, essentially. Unfortunately, the pore domain is very conserved between these channels, nearly 100% identical in amino acid sequence, making it very difficult to find blockers that are selective And what we really needed to do was find a blocker that bound outside of the pore domain onto one of these voltage sensing domains that show a lot more sequence differences between the different channels. Okay, so how do the channels work? So the voltage sensing domains, I told you that they contain these sets of arginine residues that make up the voltage sensor. And what we found, this was a paper we published in Cell in 2019 with some structural biology that we had done. We found that that this voltage sensor is able to move through the membrane field in this manner. So the voltage sensing domain, this 4-helix bundle, is designed to allow the voltage sensor to move up and down. When it does so, it pulls on this alpha helix on the lower part of the channel that is connected to the intracellular part of the pore, and it basically is able to open the gate of the channel, which is on the intracellular side. And it does this in this manner that you can see here, similarly to opening and closing the iris of a camera. Now, what I've showed you in the previous slides is the transition between the closed state of the channel and the open state of the channel. There is also an additional state called the inactivated state that occurs when the fourth voltage sensing domain becomes activated. The fourth voltage sensing domain is a little bit slower, so it takes a bit of time after the channel opens to enter the inactivated state. But this is an essential part of the function of these channels. So the channels can open and close very quickly in this manner. The channels are expressed on sensory neurons, such as these pain sensing neurons that innervate the skin and other parts of your body that are sensitive to pain. And what they do is once these cells become activated by a stimulus, these channels can very rapidly transition between these various states. And when they do so, it causes this rapid, very large change in the membrane voltage called an action potential. These action potentials last only for about a millisecond as the channels transition rapidly between these three states. And the nice thing about the action potentials is that they are able to rapidly move down axons and send signals all the way to the brain so that you feel pain. And if you are able to interrupt these action potentials, such as when you apply a drug like a local anesthetic, like Novocaine or lidocaine, you prevent the generation of these action potentials, and you can have a very strong effect on pain. And this is how we think about the Nav1.7 channels. Now, we wanted to, in order to study Nav1.7 and to determine what a drug would look like, we needed to make an animal model. And we did this before selective inhibitors of Nav1.7 existed. We wanted to ask the question, well, what would happen if we had a selective inhibitor of Nav1.7? What would it look like? And so we generated a mouse, a transgenic mouse that was basically a knockout of Nav1.7, but it was inducible by tamoxifen. So we had engineered a mouse so that the mouse could grow up and become an adult mouse with normal levels of Nav1.7 and normal pain behavior. And then what we were able to do was, if we injected tamoxifen into the mouse, we had engineered these mice to rapidly delete the Nav1.7 gene upon injection of tamoxifen. So you can see how that works here. Here we are looking at mRNA of Nav1.7 in the DRG. The DRG is the part of the body where the cell bodies of the sensory neurons are contained. So you can see that the DRG neurons express Nav1.7. Here's an expression on a log scale. When we inject tamoxifen, we very rapidly delete Nav1.7, and the RNA basically goes down to 0. This is as low as we can detect. It falls over 1,000-fold in concentration. Whereas mice that were not engineered to delete the Nav1.7 gene continue to express Nav1.7 even after injecting tamoxifen. So we can use these mice as a control. If you look at protein levels in the DRG, you can see that the protein is somewhat stable. It takes a little while. It takes actually about 2 months for the protein to reach undetectable levels after injecting tamoxifen. And again, mice that don't have the machinery to delete Nav1.7 continue to express Nav1.7 at normal levels. So we can use these mice as a control. And what we can do is basically ask, well, what happens to pain when we remove Nav1.7 and we're doing this in a very selective way. So it's kind of simulating what a highly selective Nav1.7 inhibitor would look like. And what we can see is that the mice become insensitive to multiple pain models. So here we, in the first model, we use a heat lamp to shine on the hind paw of the mouse, and we ask how long does it take to respond to this heat lamp stimulus. And we see that the mice where we've deleted Nav1.7, over time, as the Nav1.7 disappears, we see that it becomes insensitive to this heat lamp. Here's the control mice showing normal sensitivity. Here's a tail immersion model where we dip the tail of the mouse in 55-degree water bath. We ask how long does it take for it to flick out its tail. And we see that over time, the mice reach this maximum of 5 seconds. They become insensitive to the stimulus, whereas the mice that continue to express Nav1.7 are not affected. And here's a hot plate where the animals, again, become insensitive to the hot plate after the Nav1.7 gene has been removed. And otherwise, the mice seem pretty normal. We notice that they end up getting injured more easily, similar to what happens in human patients, but they look like a CIP mouse. So this pretty much validated our ability to use mice for studying Nav1.7. These are pain models that we applied to the mice after removing Nav1.7. The CFA inflammatory pain model is a very commonly used inflammatory pain model, and the Brennan model is basically a model where you introduce an incision into the paw and you sew it back up, and you ask, and that causes inflammation as well. Inflammatory pain. It's kind of a post-operative pain model. Normally, mice can feel radiant heat even at baseline, but then they become much more sensitive for a few days after this injury. Whereas the knockout mice, the Nav1.7 knockout mouse, basically does not have any feeling of this painful radiant heat stimulus at any time. So you can see that very clearly Nav1.7 is playing an important role in inflammatory pain states. And we can also, I won't have time to show it, but we've also looked at neuropathic pain states as well. And Nav1.7 is required for that as well. So the real question then was, can we make molecules or inhibitors that can selectively block Nav1.7? And we and other companies did massive high throughput screens to try to identify these molecules, to specifically look for selective inhibitors of sodium channels, including Nav1.7. And we all came across basically the same type of molecule, which is referred to as the aryl sulfonamide sodium channel blocker. So these molecules, here's an early molecule that we studied. These molecules have an aryl sulfonamide group on the right-hand side, which we call the warhead. This part of the molecule is essential for binding to sodium channels. And then we have a left-hand side that we can manipulate and help us identify molecules that are selective for particular channels. And so what you can see here is that this molecule is highly selective between different sodium channels. It blocks Nav1.7, of course, which is shown in black. Unfortunately, it blocks some other channels as well, like Nav1.2 and Nav1.6, but it has almost no effect except at extremely high concentrations on all of the other sodium channels. So this is just showing you the dose dependence of block using this compound. So this was something that we had never seen before, a molecule with an extreme level of selectivity, and this was the first time we realized that this was possible. These molecules also have the property that they like to bind to the channel in the inactivated state. So if you hold the channel in the inactivated state, the compound binds very tightly. There's a 0.1 nanomolar inhibitor. Whereas if you force the channel into the closed state, the compounds don't bind with high affinity. So they're very state-dependent blockers. So how do these molecules work? Well, we figured out here, these were some structures that we published in 2015 in Science. We found out that the aryl sulfonamides actually bind to the 4th voltage sensing domain. This is the voltage sensing domain that is involved in the process of inactivation. They bind to the extracellular part of this domain, and they interact directly with the arginine residues that make up the voltage sensor, shown here. So the negatively charged part of the aryl sulfonamide warhead, I should mention that the aryl sulfonamide warhead is negatively charged under physiological conditions, is able to interact with a salt bridge interaction with the positively charged arginine residues in the voltage sensor. And what this does is it basically prevents the voltage sensor from moving back down. It makes the voltage sensor stuck in this up position. And when that happens, the channel remains stuck in the inactivated state. The nice thing about targeting a voltage sensor, however, is that there are some residues, shown here in light blue, that are different between the different channel subtypes. And it allows us to make molecules that are highly selective. And in fact, we have now, since this time, been able to make molecules that are very specifically selective for Nav1.7. So we achieved what we set out to do. So at this point, what I want to do is tell you about the molecules that have been in clinical trials, and I want to clear up some confusion that seems to be in the field, because I read review articles and papers, and I see presentations where people talk about the inhibitors that have failed in the clinic, and a lot of them bring up molecules such as vixotrigine. This is a molecule that is an really old molecule. Was brought in. It was at one point brought in, owned by Convergence. It was eventually, which was eventually purchased by Biogen, and this has been in multiple clinical trials. But the important thing is that this is not a selective Nav1.7 inhibitor. This is in fact, vixotrigine is in fact able to inhibit all sodium channels with pretty much equal affinity. So it's a non-selective sodium channel blocker, and actually, but it has been described for some reason in the literature as a Nav1.7 inhibitor. There are other ones as well, like BIIB095 and Xenon 402. These are non-selective sodium channel blockers, and so the fact that these didn't work very well in the clinic should not be used as evidence that there's something wrong with Nav1.7 as a target, because these molecules are definitely not selective for Nav1.7. Vixotrigine is in a sense very similar to carbamazepine, which is a drug that is used in the treatment of trigeminal neuralgia. These drugs are used at low concentrations, and they have efficacy in trigeminal neuralgia to some extent, but you can't use them at high concentrations because they become toxic. The whole idea of the selective sodium Nav1.7 blocker was that we would be able to use it at high concentrations systemically and have a very strong effect on pain. These 4 molecules, in fact, are the only molecules that have only true selective Nav1.7 inhibitors that have been in clinical trials. The first one to be in a clinical trial was Pfizer's molecule, PF050817771, which we refer to as PF771. This is an aryl sulfonamide, first generation aryl sulfonamide that was in phase 1 and phase 2 clinical trials. These 2 molecules are aryl sulfonamides from Genentech. So my group was responsible for putting these molecules into the clinic. And there's an additional molecule that as far as I am aware, is still in phase 1, called ST2427. It was made by Site 1. It's actually a different kind of inhibitor. It's a saxitoxin analog. And it is binding to a different site on the channel, but it is also able to achieve a very high level of selectivity for Nav1.7. So these are the selective inhibitors that have been tested. I should mention that the Genentech molecules failed not due to efficacy, due to lack of efficacy, but they failed due to potential safety problems. They showed evidence of liver and gallbladder toxicity, and we had to abandon them after phase 1. Site1's molecule, again, is still in phase 1, so it has not yet been tested for efficacy. The only molecule, the only selective Nav1.7 inhibitor ever tested for efficacy was PF771, where it was placed into several efficacy-oriented clinical trials, including erythromelalgia, diabetic neuropathy, acute pain, and so forth. So the real question then is, if you're trying to understand why Nav1.7 inhibitors did not work, is why did Pfizer's molecule fail to show robust efficacy in the clinic? So when you, in the process of drug development, you need to be able to demonstrate that your molecules that you're making are able to inhibit the receptor that they are targeting in an in vivo setting. Just because a molecule can inhibit Nav1.7 in vitro very effectively does not necessarily mean that it's going to work in an in vivo environment. And so it's very important to make what we call PD models, or pharmacodynamic models, where we can test any particular molecule in a convenient way to determine whether it can inhibit Nav1.7 in vivo. So we constructed a mouse for this purpose. I should also mention that many of the selective inhibitors of Nav1.7 only work well on the human Nav1.7 channel. They don't necessarily inhibit the mouse channel to the same degree. So it's important to have a model with a human channel. So we basically constructed a humanized mouse that expressed the human Nav1.7 channel in the DRG neurons with a single point mutation called I848T. This mutation causes inherited erythromelalgia in humans, though in the mouse, it didn't really have much of an obvious phenotype. But what we discovered is that when we injected the plant alkaloid aconitine, which is a sodium channel activator, if we inject aconitine into the paw, we saw a very robust pain response in these mice. So basically, they flinched their paw hundreds of times in 5-minute intervals for a period of about an hour. And you can see that here. Whereas if you inject aconitine into a wild-type mouse, you have basically no phenotype. So this assay basically allowed us to determine whether we could see the activity of human Nav1.7 channels in vivo and whether our compound could block this activity. And what we found is that PF771, Pfizer's molecule, did not show any significant effect in this model. We could not demonstrate that it was able to inhibit Nav1.7 in vivo, whereas the molecules that we had developed, and we had the advantage of developing them in conjunction with this model, was we could show that we could completely block Nav1.7 in vivo with the acyl sulfonamide molecules that we had developed. They still took a lot of concentration, over 1 micromolar to inhibit the channel, despite the fact that these are picomolar inhibitors of Nav1.7. So the question is, well, why does PF771 not work in vivo? It works great in vitro. It's clearly a potent Nav1.7 inhibitor. And the reason that we believe this is happening is because PF771 is highly bound to plasma proteins in vivo. Greater than 99.99% of PF771 is bound to albumin in the blood, leaving only a tiny fraction available to block Nav1.7 channels. And it's just the amount available to block is just not enough to show target engagement in this model. Whereas these molecules are still highly protein bound, but they have enough free concentration to be able to inhibit Nav1.7 if you get them up to high concentrations. Now, and so it's important to realize that PF771 is just not able to engage the target in vivo. We have since been able to make better molecules, which I refer to as 3rd generation sodium channel blockers. We are not the only ones that have made these. Amgen and Merck have been pioneers in this area as well. Basically, these molecules contain the negatively charged sulfonamide group, as is required for these types of blockers. But also on the left-hand side, we've introduced some secondary and tertiary amines that give the left-hand side a positive charge. So these molecules are actually Zwitter-ionic compounds. And Zwitter-ionic compounds don't bind to plasma proteins as much. So a molecule like 3565 is actually about 5% free. And as a result, we can show efficacy at much lower concentrations. It has much better target engagement in vivo. And so we and others have been working on this class of molecules in more modern times. This is an example of some of the molecules that Merck developed along the same lines. They're called SSCI1 and SSCI2. And again, they are aryl sulfonamides with amines on the left-hand side, making them Zwitter-ionic compounds. And they developed a PD model as well. They used the monkey instead of a humanized mouse, but it's basically a similar type of setup. They also used odor as their stimulus. So they were able to present the monkeys with odors, monitor them by MRI, and look at these odorant responses over time. If they injected a compound, they could see that the odorant responses could be inhibited. And in fact, they demonstrated that their molecules that they had made could inhibit the responses basically to 100% if they introduce it at high enough of a dose. And interestingly, also they examined PF771. And they again, very similar to us, they found that PF771, the Pfizer compound, was not able to inhibit Nav1.7 in this in vivo environment. So really what this is showing us is that first of all, only one compound, only one selective inhibitor for Nav1.7 has ever been tested for efficacy in a clinical trial, and that molecule is not able to inhibit Nav1.7 in vivo. So it really tells us that we have not yet tested the hypothesis that inhibiting Nav1.7 channels in a human would have an analgesic effect. It's still quite possible and we expect that would be possible if we were able to do that. Another idea that has been presented in review articles and in the literature as to why Nav1.7 inhibitors have failed into the clinic is that they need to get into the spinal cord for efficacy. And in fact, there's a paper claiming that Nav1.7 is actually not involved in action potentials in the periphery, but is in fact mostly involved in the secretion of neurotransmitter in the spinal cord. So you really need to get the drugs into the spinal cord to show an effect. Now, and in fact, Pfizer's compound, PF771, is in fact peripherally restricted. It is not able to get into the spinal cord. However, the acyl sulfonamides that we developed are able to very easily cross the blood-brain barrier. On the other hand, they work pretty well. So maybe this fits with that idea. However, in more modern times, as we've made third-generation molecules like 3565, these are also highly peripherally restricted. They cannot cross the blood-brain barrier. They're Zwitter-ionic compounds, and yet they show very nice efficacy. These show better efficacy than any other inhibitors of Nav1.7 that we've ever tested, and they cannot get into the brain. And in general, we have not seen a correlation between a compound's ability to get into the brain or spinal cord and its efficacy. So we do not think that this is the problem. And just a little bit more on that, we did an experiment to kind of look at the generation of action potentials in the skin. Okay, and we wanted to know whether Nav1.7 was actually involved in this process. So we set up an assay where what we do is we measure blood flow, and it turns out that blood flow is dependent on action potentials. So the way that we do this is we use laser speckle imaging to measure blood flow in the hindpaws, and we apply mustard oil, also known as AITC, to the hindpaw. And when we apply mustard oil and we watch for this laser speckle imaging response, which is dependent on blood flow, what we find is that after a brief artifact, when we apply the AITC, we find that we see a response to AITC and not a response to mineral oil, which does not show this effect. And we know that this response is dependent on TRPA1. TRPA1 is the receptor for AITC, and it's expressed only on sensory neurons. And so we know that this is a sensory neuron dependent effect. And in fact, we know that the peak part of this response is due to the presence of action potentials that occur within the first minute of applying mustard oil. And what we discovered was that in the Nav1.7 knockout mouse, this action potential dependent part of the response is eliminated. So we know that Nav1.7 is required for the generation of action potentials in the skin, okay? Which was the original hypothesis around how Nav1.7 worked. And in fact, we can take our peripherally restricted Nav1.7 inhibitor, 3565, and very easily block this action potential dependent part, showing that clearly a peripherally restricted inhibitor can prevent the generation of action potentials in the skin. So indicating that even if there are Nav1.7 sodium channels in the spinal cord, which of course there are, a compound does not have to block those in order to show efficacy. We can get efficacy just by blocking channels in the periphery, because those peripheral channels are absolutely essential for the development of action potentials. We also did some experiments where we directly recorded sensory neurons using in vivo electrophysiology. And here what we do is we basically take an anesthetized mouse, we place an electrode either in the spinal cord to study a spinal cord neuron, or in the DRG to directly record from DRG neurons. And we can record action potentials from these cells. And so before I can show you this data, I just want to point out that there are many different types of sensory neurons. And they can be somewhat characterized by the amount of myelin around their axons. So we have A alpha and A beta fibers that have very large amounts of myelin around their axons, which means that their action potentials travel very rapidly down their axons. And then we have, on the other extreme, we have C fibers that make up the majority of the pain sensing nociceptor fibers. And these are completely unmyelinated and have very slow action potentials. And then you have these A delta fibers that have intermediate speed action potentials. Okay, so we can distinguish, just by looking at the speed of the action potentials, we can distinguish between the different types of fibers. Okay, so if we record from what's called a wide dynamic range spinal cord neuron and apply a variety of stimuli to the hindpaw, we can see signals coming from both A fibers and C fibers. So here is a recording of a spinal cord neuron, a single spinal cord neuron. We can see that the neurons respond to light touch, like camel and hog hairs, and they also respond to more harsh touch, such as pinching and high intensity von Frey hairs, and it also responds to noxious temperatures, such as 50 degree temperatures. So you can see that this neuron has both touch sensitivity and pain sensitivity, you could say. You can also stimulate the receptive field in the paw and look at the action potentials directly after a stimulation. This allows you to separate the fast-moving action potentials from the slow-moving action potentials. So here we can see after the stimulus in the beginning, we can see the A fibers and then the C fibers, and maybe some A deltas in between. And you can take this information and plot it on a raster plot where each point represents an action potential. And you can see time versus latency, how long it takes for the action potential to arrive at the electrode. So you can see again the C fibers and the A fibers in this recording over time. And what we did here is we applied a vehicle. Okay, so we applied not a drug, not an inhibitor of Nav1.7, just a vehicle that we normally put the Nav1.7 inhibitor in, and we saw basically no effect on this response. Now, if we do inject the mice with our Nav1.7 inhibitor, our selective inhibitor, what we see is that all of these responses are basically inhibited. We no longer see the high intensity mechanical responses. We no longer see the noxious heat responses. And when we look at the action potential latencies with electrical stimulation, what we find is that while injecting the compound, what we find is that the latency gets slower. So we're slowing down the C fiber action potentials. We have no effect on the A fiber action potentials, by the way. But the C fiber action potentials are slowed down and eventually eliminated. Eventually, if you wait long enough, just a few minutes, you no longer can elicit any action potentials from the C fibers, while the A fibers are functioning normally. So this tells you that the Nav1.7 inhibitor can inhibit these peripheral action potentials very effectively. And yet it cannot, they don't inhibit A fibers, which are the sensory neurons for touch, as expected, because we know that touch is not affected in the patients that lack Nav1.7 channels, the CIP individuals. Now, here is a recording that we did of directly recording from C fibers in the DRG, which we can do. You only get one fiber at a time if you're recording in the DRG. That's the advantage of the spinal cord neurons, where we can see lots of C fibers simultaneously. But here we're recording from a single C fiber, and we can see the changes in latency as shown here. And just to show that these changes in latency and the eventual loss of the action potentials is specific for Nav1.7, we generated a mouse called the modified binding site mouse, where we engineered a mutation into Nav1.7 that prevents the compounds from inhibiting the channel while allowing the channel to function normally. Okay, so this is we've removed the binding site of the compound, so the compounds can't inhibit. And when we do an experiment with DRG neurons from these mice, we see no effect. So we know that this effect on latency and the silencing of C fibers is entirely due to our compound's ability to inhibit Nav1.7. Now, another thing that I read in review articles and in papers and in presentations is this idea that the reason that Nav1.7 inhibitors have failed in the clinic is that they are not engaging the endogenous opioid system. The idea that the overexpression of endogenous opioids, in particular enkephalin, is required for the analgesic effects of Nav1.7, of removing Nav1.7. And so this never really made a lot of sense to us. It's hard to believe that that enkephalin could be driving the analgesia in the Nav1.7 knockout mouse. Because in particular, one of the things that we can do is we can dose mice with a Nav1.7 inhibitor and observe analgesia right away. We don't have to wait any period of time. So the endogenous opioid model is really a model that basically says that when you remove Nav1.7, you overexpress enkephalin in the DRG neurons. But that enkephalin needs to be, first of all, it needs to be synthesized, and then it needs to be transported into the tips of the fibers in the spinal cord, where and secreted, where it has its effect on mu-opioid receptors. But that would take some time. You wouldn't expect that an inhibitor would work so fast, even though clearly they do. But we did an additional experiment here where we looked at we recorded C fibers in the spinal cord over time. And here what we did was we added spinal morphine. So when you inject morphine into the spinal cord, you see that you can inhibit the C fiber action potentials. You can see what we are doing here. It doesn't affect the latency of the action potentials because we're not affecting the peripheral part of the nerve. We're not slowing down action potentials with morphine. We're just preventing secretion of glutamate in the spinal cord. And that can effectively silences the C fibers as well. Now, if we inject the mice with IV naloxone, this process is inhibited. Morphine no longer has its effects because we've inhibited the morphine receptor, the opioid receptor, and we recover the C fiber action potentials. If, however, we do an experiment with a Nav1.7 inhibitor where you can see that the action potentials go away and the latency increases, as I've shown before, naloxone does not recover those action potentials. So we don't believe, we've not found evidence that endogenous opioids are really playing a role in the analgesic activity of Nav1.7 inhibitors. And then we have a lot of other experiments as well that are about to be released as a publication where you can read more about that. Okay, so what I've shown you so far is that PF771 is the only Nav1.7 inhibitor that has ever been tested for efficacy, but it's not a good test because this molecule is not able to actually inhibit Nav1.7 in vivo. So then the next question is, well, now that we've made molecules that inhibit Nav1.7 very nicely in vivo, why have these not worked out? And so, and the reason is because they cause side effects. And now I'm going to switch gears and tell you a little bit about these. So with our acyl sulfonamides, we never had a chance to test them for efficacy, but we did test them for safety. That's what phase 1 is all about. And what we found is that if we dosed these molecules high enough, this is the dose in milligrams from 45 to 540. If we dose these molecules high enough and we monitored blood pressure of the patients, and each patient is represented by a black trace, the average is in blue. What we found is that once we got to about 270 milligrams, we started seeing some patients having these very low blood pressure spikes right after dosing the compound or soon after dosing the compound. And that happened only at the high dose levels, and it became more and more common at the high dose levels. And this effect was severe enough. It affected both the systolic and the diastolic blood pressure. And here below is the pulse pressure, which is the subtraction of systolic and diastolic. But this was a severe enough effect that many of the patients in this clinical trial fainted due to loss of blood pressure and had to be given IV fluids. So this was a pretty severe side effect. At the time, we didn't know whether it was on target or off target. But it definitely occurred in a dose-dependent way. It was clearly drug related. And you can see here, these are the individuals that were, the ones that got hypotension are shown in red. And you can see it's dependent on the dose. And here's the concentration that was obtained, the total concentration of the drug that was obtained in the blood. And here is basically the fraction of subjects that as you dose up, you see the more and more percentage of the patients had this hypotension effect. This was all published in 2019. So we wanted to know, is this an on-target effect or is it an off-target effect? And we discovered that mice show the same thing. So if you dose 3565, our very potent in vivo, works really well in vivo molecule, we see that when we dose this molecule, shown in the light gray here, we see a loss in blood pressure relative to dosing a vehicle. The compound does not affect the heart rate. Heart rate is pretty much the same between vehicle and the drug, but definitely we see a marked decline in the blood pressure. This was such a big effect that we were worried that it was affecting our experimental data. And of course, I just showed you a bunch of electrophysiology that we did with this compound. But we figured out a way to keep the blood pressure normal by co-injecting with phenylephrine. So we were able to balance this effect and maintain constant blood pressure when we did our experiments, so that we didn't have interference due to blood pressure. But this was a clear effect that we could see. And so we were able to ask whether this was on target or off target by taking advantage of our modified binding site mice, the mice where we had mutated the binding site of our inhibitors to try to identify what was the target causing this effect. We find in wild type mice that if you apply the drug with an IV injection, you see a decrease in blood pressure, as I showed you before. If you mutate the binding site in Nav1.6, so Nav1.6 can no longer be inhibited by this compound, even though it doesn't really get that much inhibited, because this is mostly a selective molecule, but we still see the same decrease in blood pressure. However, if we mutate the binding site in Nav1.7, we don't see this effect. So we know that this is an on-target effect. It's due to inhibition of Nav1.7. And this basically demonstrates that Nav1.7 is actually involved in the control of blood pressure. And if we block it, we have an effect on blood pressure in human patients as well as in mice. And so how is blood pressure controlled? How is this process exactly happening? We don't really fully understand, but blood pressure is controlled by the autonomic nervous system. And it's controlled by sensory neurons that that measure the blood pressure basically in the body and allow the body to maintain homeostasis using the autonomic nervous system. And these neurons actually express high levels of Nav1.7. So it makes sense as to what might be happening. We also noticed in our clinical trials of GDC 0276, these were still phase 1 trials, we noticed that we saw a preponderance or an imbalance in adverse events or AEs that looked like potentially autonomic side effects. The patients had diarrhea, abdominal pain, nausea, constipation. They even had blurred vision, which is a characteristic of inhibiting the parasympathetic part of the autonomic nervous system. So we saw these autonomic side effects when we dosed this inhibitor of Nav1.7. And of course, what we know now, we've always known this, is that the autonomic nervous system, which has 2 branches, the sympathetic and the parasympathetic branch, is involved in the homeostasis of the body. We've always known that these neurons, both parasympathetic and sympathetic, express high levels of Nav1.7. We were hoping that this would not cause us any problems because individuals that lack Nav1.7 don't have any obvious autonomic side effects. So we were hoping that even though Nav1.7 is expressed in these neurons, that it's not that important. But what we've found is that actually that is not the case. If we inhibit Nav1.7 in vivo, we have severe effects on the autonomic nervous system. This is apparent, obviously, in our human patients that we tested with a drug that was not that required really high doses to achieve in vivo target engagement. But in our more modern molecules, our Zwitter ionic 3rd generation molecules that are really good at inhibiting Nav1.7 in vivo, we found that we get really, really severe autonomic side effects in higher species, such as dogs and monkeys, even at relatively low doses. And this is what has prevented us from moving these 3rd generation molecules into the clinic. And I believe that this is our experience at Genentech. And I believe that Amgen and Merck and other people in this field have found the same problem. And this is why these better Nav1.7 inhibitors have not yet made it to clinical trials and may never make it to clinical trials because it's going to be very hard to get around this effect because it's an on-target effect. So, so in conclusion, what I've showed you is that Nav1.7 is required for pain in humans and mice. We can make selective blockers using the aryl acyl sulfonamide tools. These work by binding to the 4th voltage sensing domain, VSD4. They prevent recovery of the channel from an activation, effectively blocking Nav1.7 very nicely. We can achieve high levels of selectivity by taking advantage of sequence differences in VSD4. We can show efficacy of molecules in rodent pain models. And we know that efficacy is occurring by silencing nociceptor neurons. Unfortunately, Nav1.7 inhibitors display problems. They have problems. They have red flags in humans and in preclinical models. And we believe that it is due to on-target effects on the autonomic nervous system. That is preventing us from putting our newest generation molecules into clinical trials. And this is why we and other companies have now mostly abandoned this target as a potential pain drug target. Now, is there any hope out of this situation? There is the possibility. One thing that we noticed is that we could take a Nav1.7 inhibitor and test it, for example, in a chronic pain model, like a model of diabetic neuropathy. So in this model, we take mice. Normally, they have very little sensitivity in this particular pain model. If we make them diabetic, they become very sensitive to pain. These are the baseline animals. But if we dose this Nav1.7 inhibitor at 10 mg per kg, we see an analgesic response, right? But this is a relatively high dose and we would expect it to have autonomic side effects. However, what we found is that if we have a very low dose of the molecule, which does not show any effect on day 1, but if we continue to dose it every day, over time, on day 5 and by day 12, we start seeing the development of analgesia, meaning that even if we're inhibiting only a fraction of the Nav1.7 channels, not enough to show efficacy on day 1, but if we continue to inhibit that fraction of sodium channels over time, we might be able to observe the development of an analgesic response, which might be usable in a chronic pain situation. So we think that this is a possible approach to Nav1.7 inhibitors in the future. If people are willing to take the risks to move molecules that don't work on day 1, but move them into chronic pain clinical trials to see if they would develop this analgesic response over time. And with that, I'm going to conclude and thank my collaborators, both at Genentech, my lab, and people at Genentech that are not in my lab that have helped me tremendously with this project especially around screening and chemistry and structural biology and so forth, as well as my outside collaborators, Julian Allard, who is the amazing in vivo electrophysiologist that we've worked with, and Charlie Cohen, who was really, he was, when he was at Xenon, he was a major contributor to the thinking around this project as well. Okay, thank you. And
thank you so
open
much,
to
David.
questions.
Thank you so much, David. This was an amazing talk. We're really excited to have you and hear this amazing stories. There's a lot of questions. So we'll start with, we'll start quite a few. We might run a little bit over time if that's okay with you, because I would really like to hear your answers for some of these questions. Okay, so we'll start with a question from a colleague of mine at NCAT, Stephen Pittenger. So given pharma leaving Nav1.7 behind, are there novel targets that are of particular interest to industry? And I think you covered a lot of the second part of the questions, but I'll just ask it. Were there any specific lessons learned from the failure of Nav1.7 as pharma moves forward in the pain space?
So there are definitely other targets that we are interested in the pain space, and I am working on those today. They don't have the magic of Nav1.7 because they don't have that incredible human genetics. So they're much more, like regular drug targets that we work on all the time. We don't really know how well they're going to work. They work in animal models, but we continue to work on pain targets, no doubt about it. Lessons learned from Nav1.7. I think this is not an atypical situation. This is just part of the drug discovery process. Most of the targets that we go after are just not going to work out, and you don't really know whether that's going to be true or not until you actually go and do clinical trials and really study the target and understand it. And so I don't know if we would do anything differently if we went back in time. We always knew that Nav1.7 was expressed in the autonomic nervous system. We didn't know how that was going to affect the drug discovery projects until we were actually there seeing the effects and understanding what was going on. So yeah, it's just part of the drug discovery process, and this is why it's slow and frustrating, and you end up failing many times as a drug discoverer.
Makes sense, David. Thank you for a wonderful answer. The next question is again from Steve, and it was something I was thinking about as well, so I'll ask it. So I have heard a little bit about the inability of some of the animal pain models to translate to the clinic. Does pharma still put significant trust in these models, or are they working to augment them in different ways, maybe using advanced human tissue models? Is that something you all are thinking about?
Yeah, I mean, I think that this is a problem that plagues not just the pain space, but certainly all of neuroscience. Our models are never as good as we'd like them to be. In fact, I would say pain models are probably better than models in some other areas of neuroscience, because at least pain is something that mice experience, right? Mice don't normally get Alzheimer's disease, for example, but mice have pain very similar to our pain, but especially in studying chronic, it's very easy for us to study acute pain, I would say, in an animal model. Chronic pain is much more challenging. And in part, I think one of the big reasons is that we have to study pain differently than we would in a human. We can't talk to the mice and ask them what how much pain they're feeling, right? So we use approaches such as von Freyne hairs and heat lamps and hot plates, and in order to kind of stimulate the mice in some way and try to look for sensitivity. And that may be not the best way to study pain, but we haven't. And people have tried to look at other approaches where they're trying to study the behavior of the mice in some way to determine whether they're feeling pain. But it's a challenging area. I still think that mouse models and rat models still play a really important role in the drug discovery process. There are many cases where we have stopped developing a molecule because they don't work in the rodent models. And I think that it has saved us a lot of time and energy by not having to go into clinical trials. So it certainly allows us to weed out a lot of molecules that are most likely going to fail in the clinic because they don't even work in the animal model. But animal models are not going to be perfect. I mean, you're going to definitely see molecules that work really well in animal models that don't end up working well in the clinic. I think in the case of Nav1.7, I do think that the molecules would work in humans if we could get them at high enough levels safely. I think there's just too many similarities between the human and the mouse phenotypes with respect to Nav1.7 to think that it wouldn't work. So in this case, the models worked fine.
Thank you, David. I appreciate it. Next question here is, were all these small molecule inhibitors orally administered? Other than high plasma protein binding, did they have optimal PK characteristics, clearance, hepatic microsomal ability, permeability, question mark?
Yeah, so obviously our goal and other companies' goal as well was to find orally available drugs. So that's what we went after. So the aryl sulfonamides are orally available. I mean, you can dose them orally. Certainly in all of our behavior experiments that we show, and many of the experiments, it was dosed orally. The only time we dosed the molecules IV is, for example, in vivo electrophysiology, where the mice is under anesthesia. So we don't have the ability to dose them orally. So we dose them IV, which can also be done. But the drugs are meant to be orally dosed. And they do have excellent, I mean, obviously you can find many molecules that have very poor PK properties, but we and others have certainly been able to find molecules with excellent PK properties. And in fact, in the most recent molecules, we can find them that have significant free fractions as well. So I would say that the pharmacokinetic properties of the molecules are excellent. That is not the problem. Yeah.
Fantastic. There's plenty of questions, David, so I'm going to keep going, but
Sure.
You can tell me if, So next question here is, can you comment on the location of these Nav1.7 channels, and are they only located peripherally? You might have touched on this, but, I'll ask you again.
So research that has been published where people have used antibodies to stain for Nav1.7 in vivo have clearly demonstrated that the channel is present throughout this sensory neuron. So it's present in the cell body, it's present in the peripheral fibers in the skin, and it's present in the at the central end of the sensory neurons as well. So it's, as far as we can tell, it's expressed throughout the sensory neuron cell, and it's not really clear. There's not been any convincing data yet, at least, to suggest that it's more highly expressed in one part than another. Yeah.
Thank you, David. Next question. Based on your experience and that of pharma generally, is having a good inducible animal model of the target essential before initiating medchem? Also, is it typical for independent large HTS to find the same warhead for a target?
Yeah. Okay, so the first question, I think inducible knockout mice are fantastic. Obviously, it only works in a case where you want to block the receptor. So sometimes we want to enhance the activity of a receptor. So it's not as easy to make such a model. But it basically gives you a chance to see what a molecule would look like if you were able to develop one. I think that's a really valuable exercise. And it can save you a lot of time and money, right? Because it took us years and years to get to the point where we had Nav1.7, selective Nav1.7 inhibitors that were good enough to test in vivo, right? That had good enough PK properties, low enough protein binding, good enough selectivity. It takes years and years to do that. And it takes a huge team of chemists to make that happen. So you spend a lot of money. If you can, early on in a project, if you can make an inducible knockout and look at what a compound would do if you were able to make one, and if you decided that, that data suggested that the molecule wouldn't do what you want, then you can save a lot of time and money and effort, and not have to go through the whole process of making the molecules. So I think it's a very useful thing. I don't think it's essential. There are certainly many cases where people very early on have a molecule that works and shows efficacy, and you don't have to go through the process of making the knockout. Certainly, if it's really easy to make a molecule that works well in vivo early on in the project, then you can use that instead, right? So the second, I've already forgotten the second part of the question.
Well, let me find it. All right, where is it at? I think Abigail moved it to answer it. Let me find it. I'm sorry.
Is it typical for independent large HTS to find the same warhead for a target?
It's very typical. One of the reasons for that, I think one reason is that these compounds are very rare. So the aryl sulfonamides are just a really special group of compounds. There's no doubt about it. I haven't seen. It's rare to find another type of molecule that can do the same thing that's not an aryl sulfonamide. Another thing is that it is not the case that all of our screening libraries, so each company will have a library of compounds that they use for screening that may contain like several million compounds, but they're not independent of each other, right? We are definitely screening the same, a lot of the compounds are basically the same compounds. And everybody is screening the same set of compounds. And each company will have their own unique set as well. But I would say that the majority of the compounds are common between the chemical libraries of different pharmaceutical companies. So you expect to find many of the same things.
Thank you, David. Okay, so I'll take the ones that are voted with the attendees, the questions. So I'll take this one. Would central administration of agents like ASOs, peptides, and antibodies avoid effects on the autonomic nervous system?
Yeah. So if you were able to, first of all, we don't know for sure that blocking Nav1.7 at the central side would be effective. We know that that the compounds are effective if we block them in the periphery. We don't know for sure whether they would work if they were applied on the central side, but let's assume that they would. I mean, certainly an ASO. If you injected an ASO centrally, you should be able to downregulate Nav1.7 only from certain neurons that are present in the central nervous system. Of course, autonomic neurons are also present in the central nervous system, but we've found an interesting effect where I suspect that the ASO would not cause that problem. which is that if you block Nav1.7 very slowly, for example, we can do that with our inducible knockout mouse. It takes 2 months for Nav1.7 to completely be removed. We don't see any autonomic side effects under those conditions. Those mice have normal blood pressure, for example. So I think it's possible that that if you block Nav1.7 slowly enough, like with an ASO, that an ASO would also be a very slow. It would take several, like a month or so to remove Nav1.7 with an ASO, because ASO is targeting the RNA, not the protein. I would suspect that you would avoid autonomic side effects if you could do that. It's just the big problem is making an ASO that's convenient enough to be used by patients, right? I think that's the big barrier there. But I think that it would work.
Thank you, David. Maybe 1 or 2 more questions and we'll let you go. This has just been really very, very interesting. So everyone has a lot of questions. So I'm taking the most highly voted questions. So anyone on the line that, would like to vote those questions, go ahead and do so. Can any in vitro safety testing foresee the inhibitor's side effect on blood pressure?
In other words, did we pick up the effect on blood pressure with preclinical experiments? Is that?
Yeah, I mean, they're wondering whether anything in vitro that could have been done. I mean, I can't think of anything, but yeah.
Yeah, we did not see an effect in any of our in vitro. We do in vitro kind of cardiac smooth muscle cell experiments, for example, but we did not see. But those cells are don't express Nav1.7, first of all, and there is no the autonomic autonomic nervous system is not there when you're doing in vitro assays. I think it's very difficult to have an in vitro assay of the autonomic nervous system effect. Yeah, so we would not have seen that. We did see it in preclinical models. But we considered it, a lot of times you'll see things, even before you go into the clinic, and you still want to go into the clinic because you don't really know whether, to what extent that effect is going to, show up in a clinical situation, right? As long as the effect that you're observing is monitorable and reversible, often you'll try it out in the clinic anyways. But we were able to see effects even in dogs, for example, before we went into the clinic.
I think they were asking on the side effect on blood pressure, particularly. So
yeah, that's right. We saw effects on blood pressure preclinically. They didn't seem to be that severe. They seemed more severe in the patients. I should say that they only occurred in patients when we dosed the compound as a cyclodextrin elixir. So in other words, there we could achieve really rapid absorption of the inhibitor. And that's where we saw the blood pressure effects. We find that if we have a more slowly absorbed formulation, we don't see as much of an effect. So, and we understood that. So we thought that maybe the blood pressure effect would not be a serious impediment to the development of our compounds. I think the biggest problem was the autonomic side effects that we ran into in preclinical animal, like in the dog. The dog just has really, really severe autonomic side effects. It makes it very hard to convince your safety group that the molecule is safe enough to test in humans, and so that has been our biggest impediment.
Thank you, David. We'll take 1 more question. We're 15 over, and it sounds that there's a lot of interest from here. So we'll invite you again at some point and we'll cover more questions. So the last question here that we're going to take is, what do you think is driving the divergence in blood pressure regulation between the human genetics and pharmacological inhibition? There's multiple questions here and you can just answer what you can. Is there compensations that occurs during development in individuals with LOF mutations, or do they also have some amount of misregulated blood pressure?
Yeah, so good questions. I have not heard reports of misregulated blood pressure in the CIP individuals. They seem like for example, you might think that orthostatic hypotension might occur in the absence of Nav1.7, but we haven't seen it, or it's not a reported phenotype of those individuals. I suspect that the autonomic nervous system is able to adapt in the absence of Nav1.7 over time. And in fact, even when we've seen the effect in patients, it's a fairly short effect, right? It happens, it's a spike that happens right after dosing with the elixir formulation that is rapidly absorbed. And then over time, the patients recover, and yet they still have the Nav1.7 inhibitor is still present. So I suspect that the autonomic nervous system is able to adapt over time. And that may be a way around the problem, potentially, because it seems that the pain sensing function of Nav1.7 is not able to be compensated in CIP patients or in mice that lack Nav1.7, right? In other words, the sensory nervous system seems to continuously require Nav1.7, whereas in the autonomic nervous system, if you remove it, it seems that it can recover, probably because of the expression of other ion channels there in some way. And of course, the autonomic nervous system is designed to maintain homeostasis. It's designed to recover from, when you push it in one way, it'll push back in the other way. So I feel like it makes some sense that the effects would go away over time when you continuously dose a patient. And it kind of continues to give ideas around, if you dose the compound very slowly, kind of ramped up the dosage, you might be able to avoid the autonomic effects. That's something that we never tried to do at Genentech. We thought it was just too risky of an approach, and our molecules just showed too much safety problems in preclinical species to allow us to be able to put them forward. But it's possible that a company that was willing to take some risks might be able to try dosing a compound much more slowly in the clinic and kind of build up a dose, and maybe that would avoid the problem.
Thank you so much, David, for this amazing presentation, amazing discussion. We really, really enjoyed having you here. I appreciate the time that you spent with us today, and we recorded this, so this amazing resource of a talk will be also available for the community to learn from. So thank you so much again. Thank you for David being here today. Thank you everyone for attending. We look forward to seeing you all in our next webinar as well. You all have a great day.
Thank you so much for inviting me. It was a pleasure. And have a great day, everybody.
Thank you. Bye, everyone.